The second sequence of PowerPoint notes for Unit 2 will be an overview of the major eukaryotic cell structures. This information is coming primarily out of chapters 4 and 5. We'll actually start with chapter 5, which is an overview of the plasma membrane and molecular transport, and then back up and pick up information from chapter 4, which covers the major organelles of the eukaryotic cell. So here's the chapter numbers and also the suggested reading for each of the two chapters. So we've been talking quite a little bit about the two basic types of cells. We know prokaryotic and eukaryotic cells are the two cell lines, and we've talked some with regards to the evolutionary history and origins of life and of these two major types of cells. In the 1800s, compound light microscopes had developed to a point where we were able to start to get a little bit better look at living organisms at the microscopic level. And due to this improvement in technology, we actually started to understand there were some fundamental connections that all living organisms shared. And there were two individuals, Schleiden and Swan, who proposed basically the first two tenets of the cell theory today, which say all living organisms are composed of one or more cells. And then the second major tenet is that the cell is a structural and functional unit of life. So structural means that living organisms are made up of cells, and functional means that these cells are the location where the chemical reactions occur that support life are happening. A little bit later in the 1850s, we get the third tenet from the cell theory from Virchow, which basically said that cells come only from pre-existing cells. When we take a look at the structure of eukaryotic cells, we see that there are basically three major anatomical subdivisions. Those include the plasma membrane, basically the membrane that surrounds the cell. Its primary function is to control what comes into and out of the cell. We'll spend quite a bit of time looking at the plasma membrane and the functions of the membrane. The cytoplasm is essentially the water-based fluid on the interior of the cell, where the organelles, these specialized structures that do specific functions for the cell, are found. And so the eukaryotic cell, three major anatomical subdivisions: the plasma membrane, the cytoplasm, and the organelles. We'll spend quite a bit of time on the plasma membrane and then come back and talk about the major organelles of the eukaryotic cell. If you look at the graphic on the left, you can see the outline of the cell. That basically is the plasma membrane, and then that dark oval structure is a nucleus, and all of that area between the nucleus and the plasma membrane is the cytoplasm. That's the location or where you would find the organelles for eukaryotic cells. The outer membrane of the cell basically has been described as selectively permeable. And if you take a look at the graphic to the left, you can kind of see the basic structure of the plasma membrane. Again, its major function is to control what comes into and out of the cell. And so the membrane provides this kind of protective barrier and helps to maintain these relatively constant internal conditions inside of the cell, these optimal conditions for chemical reactions to occur in. Those process is called homeostasis. When you look at the membrane, you can see it's composed up of two layers, basically phospholipids, and then arranged in this phospholipid bilayer are these series of proteins that are kind of randomly arranged throughout that phospholipid bilayer. Some cells, plant and fungal cells, in addition to the plasma membrane, will have an outer structure that provides additional structure and support to the cell, which is called the cell wall. You won't find the cell wall in animal cells. The cytoplasm is the water-based solution on the interior of the cell, and we'll talk at length about what a solution is, what it's made up of, and really look at the cytoplasm and the extracellular fluid being separated by the plasma membrane, and the role that the membrane has in terms of controlling what comes into and out of the cell. One term you need to know related to the cytoplasm is cyclosis. Cyclosis refers to cytoplasmic streaming, and so the cytoplasm will actually move or circulate within the cell. This helps to distribute chemical compounds throughout the cell in terms of distributing nutrients, things like glucose and amino acids, and to the various organelles that need them for their specific function. But it also helps in terms of like eliminating waste products from the cell. So that cytoplasmic streaming is called cyclosis. The major organelles of the eukaryotic cell are one of the things that separate it from prokaryotic cells. And so prokaryotic cells in general perform most of the functions for the cell on the inner membrane of the cell, whereas eukaryotic cells have a division of labor. And then sometimes they've described the eukaryotic cells as being like a factory, with each one of these organelles performing a specific function for the cell. Usually these organelles are separated from the cytoplasm by some type of membrane. The two exceptions that I can think of basically are the ribosomes and the nucleolus. But other than that, the majority of these organelles are kind of a membranous structure. And again, each one of these organelles will perform a specific function for the cell. These are primarily chemical reactions, and we'll look at each one of those organelles as we get further into this unit. This is a little bit closer look at a plant cell. The earlier graphic showed a comparison of animal versus plant cells. Similar in a lot of ways, but if you look closely, you can see that the plant cell has a few things that the animal cell did not, which include the chloroplast, which we know contains the chlorophyll and is a site or location for photosynthesis. And then in addition to the plasma membrane, in plant cells you have this outer structure, which is the cell wall, composed of cellulose, which is basically a polysaccharide, just in essentially these repetitive sequences of glucose molecules. And then all of the organelles, some of them that you I'm sure are familiar with, the mitochondria, the Golgi apparatus, vacuoles, ribosomes, and those are the organelles that we'll discuss later in this section. The major functions of the cell, remember that the plasma membrane is responsible for controlling what comes into and out of the cell. The organelles will do specific functions for the cell. And then the DNA is kind of the brains of the operation. Essentially it provides a way to store and copy and transfer information, and the DNA basically controls heredity and cell division and protein synthesis. Let's look closer at the actual structure of the plasma membrane. And the plasma membrane has been described as a fluid mosaic model, and the fluid component of this description really refers to the fact that the surface of the membrane, both on the outside of the cell where you have the extracellular fluid and the inside of the cell where you have the cytoplasm, there will be a layer of water kind of up against or right next to the surface of the membrane on both sides. That gives that membrane a fluidity and allows those phospholipids actually to move within the membrane, and also the globular proteins to a certain extent. Mosaic, if you think about art, a mosaic is really kind of this combination of pieces, and sometimes they're arranged in somewhat of a random order. And so the mosaic refers to these globular proteins that are randomly arranged in this phospholipid bilayer. When we hear the term globular protein, remember that proteins are made up of amino acids, and proteins can have different structures or levels of conformations, and the primary structure of a protein is just a linear chain of amino acids. A globular protein has a little bit more complex structure in that you take that linear chain of amino acids and fold it or twist it or coil it into a three-dimensional shape. The model piece of the fluid mosaic model refers to basically the understanding that we are continually bringing in new information and starting to really develop a better appreciation for how this membrane actually works and what each one of these globular proteins do in terms of providing support for the cell. So that's the fluid mosaic model of the plasma membrane. We'll actually do a drawing, and there will be a video of this drawing that will be posted to Canvas that will be available to you that will give you a little bit more detail in terms of the structure and the function of the plasma membrane. The fluid mosaic model basically describes a membrane that is composed of a phospholipid bilayer with globular proteins randomly arranged throughout. The phospholipids are amphipathic molecules. Essentially, this means that different portions of the molecule will react differently to water. The head region, which is essentially a phosphate group, is what they describe as hydrophilic. It means water loving. And so that component or that portion of the molecule will actually attract water. The tail component or the tails of the phospholipid are made up of lipid molecules, and lipids are like oils, and if you think about how oil and water get along, not so well, right? And so the tail portion of the phospholipid bilayer is hydrophobic, and so those tails will actually repel water. What it does in a practical sense is to ensure that you have water basically on the surface of both sides of the membrane, but you don't get a lot of water that's moving back and forth across the membrane between these phospholipids. Water will move, but when water moves, it moves through these globular proteins, and we'll see that process as we look at channel globular proteins and molecular transport in a little bit more detail. There are basically two major groups or types of globular proteins. There's peripheral globular proteins and integral globular proteins. The peripheral globular proteins, if you think peripheral, these are things that are kind of on the outside looking in. And so the peripheral globular proteins basically are just attached to the surface of the membrane. They don't penetrate both layers of the phospholipid bilayer. And you'll find the peripheral globular proteins on both sides of the membrane, on the extracellular side and also on the cytoplasm side. Their function essentially is to provide structure and support to both sides of the membrane. The integral globular proteins penetrate both layers of the phospholipid bilayer, and we'll spend the majority of our time focusing on those integral globular proteins. If you see integral, think integrated, and so these are globular proteins that are basically integrated or embedded in both sides of the phospholipid bilayer. There are literally hundreds of these integral globular proteins, and we're still learning more about the specific functions of all of these different globular proteins, these integral globular proteins. We're going to try to keep it simple and break it down. We'll do 5, but the 5 really fall into a couple of these different categories. We'll look at transport globular proteins, which will include channel globular proteins and carrier globular proteins. We'll also look at globular proteins that are involved in cell recognition in terms of identifying the cells belonging to the individual. And then we will look at a certain type of globular protein, which we will call a receptor globular protein, that binds to or interacts with certain types of molecules, basically hormones and enzymes that promote or turn on chemical reactions inside the cell. The first globular protein that we'll talk about, or integral globular protein that we'll talk about, is a cell recognition globular protein. And if you look at this, the identifying trait or characteristic for this particular protein is a unique carbohydrate chain that's attached to the surface. And so your cells, all living organisms, their cells have these globular proteins, cell recognition globular proteins, embedded in their membranes of their cells. And the carbohydrate chains that are attached to those globular proteins, sometimes they'll call them a glycoprotein, basically are unique to that individual. And that allows this almost like a fingerprint, and provides that organism the ability to identify that cell as belonging to that individual. And it's associated with your immune response. And so we have cells in our immune response system that will basically look at these unique carbohydrate chains and say, oh, this particular cell belongs to this individual. Or sometimes, if we have a foreign invading cell, like a bacteria or maybe even a virus, if that cell is recognized as not belonging to the individual, it can trigger an immune response by your white blood cells, which will basically attack and break down those foreign or invading cells. This can be an issue for people who are undergoing tissue or organ transplants, and so it's important that when those types of procedures are done, that they take measures to help ensure that the tissue or the organs aren't rejected based on these, a match or not a match with respect to these unique carbohydrate chains on the surface of these cells. The next integral globular protein that we'll discuss is the receptor globular protein. And the receptor globular protein has a binding site, and that binding site is it's a lock and key interaction with certain types of molecules, and those molecules are hormones and enzymes. And so those hormones or enzymes will attach to that binding site, and then they will send a chemical signal basically to elicit a response from the cell. So for example, some of you may be familiar with people who have trouble balancing glucose or blood sugar levels, maybe because they don't produce insulin or insulin isn't recognized by the cells. But insulin would be an example of a type of molecule that would attach to this binding site that would tell the cell, hey, we need to start to take up some of this and store some of this additional glucose that's in the extracellular fluid. And so things like insulin and adrenaline would be examples of hormones that might interact with this binding site on these receptor globular proteins and then signal essentially the cell to perform a specific function. Some molecules, remember the primary function of the plasma membrane is to control what comes into and out of the cell. But some molecules can freely cross basically the plasma membrane through the phospholipid bilayer based just purely on the concentration gradient. And so molecules will tend to move from a higher concentration to a lower concentration seeking equilibrium. That process is called diffusion. And some molecules, if they are nonpolar, can freely move across the plasma membrane through the phospholipid bilayer. And examples of those types of molecules might be hydrocarbons or oxygen and carbon dioxide. We know those are important components of cellular respiration, and so that's how those gases are exchanged across the membrane. Some molecules can't move freely through the phospholipid bilayer, and when you see those terms nonpolar and polar, that really is basically a description of whether or not the molecule has a charge associated with it. So nonpolar, basically these are molecules that don't have a charge associated with them. They can move freely across the phospholipid bilayer. Polar molecules have a charge, and so if they have a charge, even if it is a partial charge like water, those molecules can't freely move through the phospholipid bilayer. So when they will move through one of these globular proteins, one of these transport proteins. And those transport proteins could include like channel globular proteins, carrier globular proteins, and the The channel globular protein and the first carrier globular protein that we will look at are involved in a type of molecular transport called facilitated diffusion. In facilitated diffusion, the proteins are helping to move a molecule back and forth across the membrane, but the molecules still follow the concentration gradient. That is, they are still going to move from a higher concentration to a lower concentration, seeking equilibrium. And so types of molecules that move this way might be water and or glucose. Now, once in a while, the cell will actually move a molecule against the concentration gradient, and a lot of times these are ions, and so these are basically charged elements or molecules that the cell will invest energy to move and pump those molecules basically uphill or upstream. And so they'll move those molecules from an area of lower concentration to higher concentration in an effort to kind of almost like establish an electrochemical gradient. It's a way for the cell to kind of store basically an energy source that it can use to fire or trigger a specific response or chemical reaction. The sodium potassium pump is one that is an example of molecules that are moved under these types of processes. And so the cell uses energy in the form of ATP to move those molecules against the concentration gradient. That's called active transport. That occurs through the carrier globular protein 2. So this basically is what we've been talking about in the earlier slide. Channel proteins form a channel or a conduit that allow molecules to move across the membrane, and this is called facilitated diffusion. Examples of molecules that move this way would be water and or glucose. Water moves through a specific type of channel globular protein called an aquaporin, and the movement of water from a higher concentration to a lower concentration seeking equilibrium is a type of diffusion, but it's a specific type of diffusion called osmosis. Glucose is another one. It moves in a little bit different fashion in that it will move through a carrier globular protein that actually changes shape. It's still following the concentration gradient higher to lower. There's no energy investment by the cell, but that first carrier globular protein that we discussed is essentially just helping to move that molecule across the membrane based on a shape change. Both of these are types of passive movement, and so there's no energy investment by the cell, and the molecules that are moving are following the concentration gradient from a higher concentration to a lower concentration seeking equilibrium. So this again is a bit of a summary here of diffusion, and it's simply molecules moving from a higher to a lower concentration seeking equilibrium. And most of us have experienced this in some form or fashion. So if you've ever had an encounter with a skunk or someone that's wearing a lot of perfume, you'll notice that those molecules are more concentrated in one area, and gradually those molecules are moving from that area of higher concentration to lower concentration seeking equilibrium. The same process happens across the plasma membrane in terms of molecules moving into and out of your cells based on those concentration gradients. It's higher to lower and passive process with no energy investment from the cell. Another type of carrier globular protein that will do a different type of molecular transport is what we will refer to as carrier globular protein 2. And this particular carrier globular protein will actually use energy to move molecules from an area of lower concentration to an area of higher concentration. So it's actually kind of building basically a higher concentration of molecules across the plasma membrane between the extracellular fluid and the cytoplasm. And so this process is called active transport, and it uses energy in the form of ATP. A couple of ions that are moved in this fashion are sodium and potassium. When the cell does this, you actually have a lower concentration of sodium ions inside the cell in the cytoplasm, and a higher concentration of sodium ions outside the cell in the extracellular fluid, and the cell will invest energy to push those sodium ions from that lower concentration to the higher concentration. And in a similar process, it will also use those same carrier globular proteins to move potassium. And so if you have a lower concentration of potassium outside of the cell in the extracellular fluid and a higher concentration of potassium inside the cell in the cytoplasm, that potassium will be pushed from a lower concentration to a higher concentration using energy in the form of ATP. What ultimately ends up happening is you develop this electrochemical gradient, which is a kind of a stored energy. And some proteins can allow those ions to rush back through or across that membrane. When they do that, they release an electrical impulse. And so your nerve cells will do this in a way to signal or send basically an electrical stimulus to your muscles to respond. And so that's called the sodium potassium pump. And that's an example of that active transport through this carrier globular protein 2. The little more specific detail in terms of the components of diffusion, and when we're talking about molecules moving, we're really talking about them moving in these water-based solutions on the inside and the outside of the cell. On the inside, the cytoplasm is a solution, and on the outside, the solution is the extracellular fluid. When we consider these solutions, we really need to think about the components that make up the solutions. And a solution is made up of two things. It's made up of a solute, usually a solid or gas, for example things like oxygen, carbon dioxide, glucose, and the solvent. And for our purposes, the solvent in our solutions we'll always just consider to be water. So it's a combination of those things, the solute and the solvent, that make up the solution. And those two things, the concentrations of those two elements of the solution, the solute and the solvent, always have to add up to 100%. And we'll look at some concentration gradients here in just a little bit and think about how those concentration gradients affect the movement of molecules through these processes of diffusion and osmosis primarily, and really consider what the effect that may have on the cell in terms of volume and pressure. So the graphic to the left basically just shows dye molecules moving from a higher concentration to a lower concentration, seeking equilibrium across a semi-permeable membrane. The little dotted line essentially represents that semi-permeable membrane, the phospholipid bilayer, and the globular proteins. Down below it shows you might even think of this in terms of like the movement of oxygen and carbon dioxide across that membrane. One of the other things to be aware of is the solute molecules, depending on the characteristics of the solute, can move back and forth based on the concentration gradient, but also the solvent, the water, is going to move through that process of osmosis. So osmosis is a specific type of diffusion that involves water, but it's still going to be a passive process in that it doesn't use energy to move the molecules. The water molecules are just moving based on the concentration gradient from an area of higher concentration to lower concentration, seeking equilibrium. And you can see in this tube we have again one of these little membranes that's separating essentially the two sides of the tube, and on one side of the tube we have a higher concentration of a polysaccharide or a sugar molecule and a lower concentration on the other. And so as you look at this, you have a lower concentration of the sugar molecule on the left-hand side of the tube, higher concentration on the right-hand side. The tendency would be for those sugar molecules, that polysaccharide, let's say it's starch, which is a large molecule, would be to move from a higher concentration to a lower concentration, seeking equilibrium. But if those molecules are too large, or if they have a charge associated with them, they cannot cross that membrane. So because the solute molecules can't move, the water molecules are going to move. And if you think inherently, if there are fewer sugar molecules on the left hand side, that means that there's more room for more water, and so there's a higher concentration of water actually on the left hand side of the tube than the right hand side, and that water is going to move from an area of higher concentration to lower concentration, seeking equilibrium. Now because the tube is open, it doesn't create a pressure problem inside of the tube. But if this was a closed system like a cell, as that water was moving into the cell, it would eventually create a volume and pressure problem for the cell. When water moves across that plasma membrane, remember it's going to move through those channel globular proteins, and specifically the name for the channel globular protein that water moves through is called the aquaporin. It is essentially a type or a process of facilitated diffusion. So that brings us to some of the issues that cells can have with respect to volume and pressure based on the movement of the solute and or the solvent components of the solution. If you suspend a cell in what's called an isotonic solution, the cell really doesn't experience any problems or any effects with volume and pressure. Iso means the same, and so essentially the solute and the water concentrations are in balance across the membrane, in the cytoplasm and in the extracellular fluid. So the cell really doesn't have an issue in terms of volume or pressure. When you look at these prefixes and think about solutions, these prefixes are describing essentially the concentration of the solute in the solution in the extracellular fluid. And so if you see isotonic, basically that means that the concentration of the solute molecules is the same both in the extracellular fluid and in the cytoplasm. No problem in terms of volume and pressure in the cell. The other prefix is hypo and hyper you might be familiar with. Hypo means lower. And one of the things, really important pieces to take away from this, is that those prefixes are describing the concentration of the solute in the extracellular fluid. Hypo means lower. Okay, tonic means concentration. So hypotonic, a hypotonic solution means that you have a lower concentration of the solute in that solution in the extracellular fluid. If you have a lower concentration of the solute, that means you have a higher concentration of the solvent being the water. So if you suspend a cell in a hypotonic solution, essentially you have a higher concentration of water outside the cell in the extracellular fluid, a lower concentration of water inside the cell in the cytoplasm. That water is going to go from a higher concentration to a lower concentration, so it's going to move into the cell, causing the cells to become hydrated. But if a cell remains in a hypotonic solution for an extended period of time, eventually as it gains more and more water, you're going to increase volume and eventually pressure inside of the cell, and the cell can actually rupture. So that's why it's important when you get say if you're extremely dehydrated and they need to give you basically an IV to rehydrate you, they're going to give you a solution that's isotonic or in balance with what your normal concentration of your cytoplasm would be, so that you don't experience those issues. You can still get hydrated without experiencing an issue with volume and pressure inside of the cell. If we reverse that scenario and we suspend a cell in a hypertonic solution, Most of you have heard hyper. Hyper means high or above, and so like a hyperactive kid has a lot of energy. Now you remember that prefix hyper is referring to the concentration of the solute in the extracellular fluid. And so if you have a more solute, that means that you have less solvent. And so if you have less solvent, that means less water outside of the cell, more water inside of the cell. And so that, again, that water is going to go from a higher concentration from the cytoplasm inside of the cell to the extracellular fluid. And the cell is basically going to be losing water. It's going to become dehydrated, and so the volume and the pressure inside of the cell is actually going to decrease. And that process is called crenation. And you can see that especially like in plant cells. If a plant doesn't have an adequate water supply or water inside of the cell, then it loses volume and pressure, and it loses that ability to maintain what's called turgor pressure, which is that pressure that pushes the membrane out away from the cytoplasm up against the cell wall. And so as it loses that water through that process of dehydration and crenation, the cell will actually kind of shrivel and shrink. So there will be some examples of these concentration gradients done in a video that will be made available to you in Canvas, and that video will be called diffusion osmosis examples. Would really encourage you to take a look at that. It'll help you to visualize, I think, and apply these concepts of iso, hypo, and hypertonic solutions and the movement, molecular transport, and the movement of molecules, the solute and the solvent in these different solutions. So that kind of wraps this up on the plasma membrane. We'll come back and finish up with an overview of the major eukaryotic cell structures. We will use study guide worksheet number 2, not so much PowerPoint notes, to cover the information over the eukaryotic cell structures.